Okay, so in this video, we're going to look at statistics and how we can apply them in biology. So when we talk about like graphing our averages or our means, sometimes when we take like samples and we find their average, that is the mean. And we oftentimes will use bar graphs to show that mean, but sometimes just because you calculate an average, sure, you know what the average height or growth or any, like whatever the thing is, you know the average, but what does that actually tell you about all of the data points that went into finding that average? So we're going to use error bars in order to show the variability of the data. So if you have your mean or your average in a standard error bar, in a bar graph, like you know the average, but then these bars that we add basically are showing the like the variation around the mean, like how many data points, like what is their, kind of like their spread to find that average. So here you can see in the red graph how those data points are more spread out, so those bars are a little bit bigger, and then in the blue one, the data points are closer to the average, so the bars are smaller. Now this is a super simplified example, but at least it shows a visual of what we're talking about. So we have two formulas we're basically going to talk about. We have our standard deviation as well as our standard error of the mean. So with the standard deviation, what we're talking about is if you have your mean, how much does your data or all the numbers you collected like deviate from the mean? So when we talk about standard deviation, we talk about plus or minus 1 or 2 standard deviations of the mean. So if you have your mean or your average and we say plus 1 standard deviation or minus 1 standard deviation, what that means is you go up 1 standard deviation above the mean and then below 1 standard deviation of the mean. And with that, 68% of your data would fall within that range. So if you sampled 100 frogs and their length of their legs, 68 of those 100 frogs' leg lengths would fall within this range. So you have your average, but then 68% fall within plus or minus 1 standard deviation of the mean. Whereas 2 standard deviations of the mean, plus or minus, would fit 95% of your data. So 95% of those frogs would fall within that range. So if the frog legs were all very like similar, you would have a very small standard deviation because it'd all be very close to the average. But if you had lots of variation in those frog legs, and some were really long, some were really short, you would have like a larger standard deviation to explain how all those frogs are kind of spread out. So here we have two groups of corn. When I look at group 1 versus group 2, I can see that group 1, there's less variation in the corn heights, whereas in group 2, there's more variation in the corn heights. So if I found the average for each of those, and I wanted to calculate the standard deviation, you would see in group 1, you would have a smaller standard deviation because more of the sample, like the corn, they're all very close together. So 68% would fall within a smaller range, and then 95% would fall like pretty close to each other. Versus in group 2, when you had lots of different heights of corn, you find that your standard deviation is going to be larger because there's more variation in our data. So to show 95% of your corn sample with some very tall, some very short, your standard deviation would be greater. Okay, so this is helpful because when you look at just the average, it could very well be that group 2 and group 1 calculate to have the same average height. However, if you only had bar graphs, you would think those two groups were the same. But when you add the standard deviation to your graph, you can see how in group 1, the standard deviation is smaller, so therefore that tells us that more of the sample was close to the average. When you look at group 2, with it being a larger standard deviation bars, you can see that that means there's more variation in our data to get to that average. So that's helpful when you're looking at results of experiments and things. You can kind of make inferences about that sample based on these error bars. Okay. So sometimes you can see here, this one has a very large, this one is standard error of the mean versus small. And right now looking at that, you can make inferences about the sample that went into calculating those bars. All right. So, but if I do a sample, let's say I collect data on those 100 frogs and I measure leg lengths. But I'm curious, like, how does my actual 100 frogs average compare to the real frog population in all of Panama or Costa Rica or somewhere, right? Like, how accurate is my sample to represent the actual mean? We're going to use something called the standard error of the mean. So in this, like, how close is our sample mean to the actual mean is really what standard error of the mean means. And so one thing that is important in your accuracy and how close your sample actually represents the real population is your sample size. So if you're buying like here I have vacuums as my example, they both have 4.5 stars, but the one with 9,000 reviews is more accurate, more trustworthy, and more reliable. So the larger your sample size is, the more accurate your results will be. So when we look at the formula for standard error of the mean, we find that it has two parts. It has the standard deviation is at the top. That's what the S is. And then it also has, let's see, at the bottom it has the square root of the sample size. So when we talk about standard error of the mean, there are two things that can cause or influence the error bars. One is the standard deviation. If you have a lot of variation in your data, your S, your standard deviation, is going to be large, and therefore your standard error will also be large. So if we go back to this, right here, this is a standard error of the mean, and this is actually showing, like with these large error bars, this is showing us there's lots of variation in the mean number of spots per adult male guppy. Like that is a large standard deviation creating that size of error bars. And then here, well, on the shorter one, it would be like there's less variation, there's less standard deviation, so you have a smaller standard error of the mean. But your sample size also plays a role. So if you are basing your conclusions on a very small amount of data, then you could also have a large standard error simply because it's not super reliable. You only had like 10 fish or 10 people or 5 frogs or something. And so therefore you're like, I'm not really sure. Like I'm not very confident with what I'm saying because I'm based on a small sample size. So when you look at like what it means, like these two standard error bars, in sample 1, that could be either because sample 1 had large variation, and so therefore you are saying that you are 95% confident that, like if we look at this, you are 95% confident that the real population average or mean falls within this range. And so that is what this is saying. So this either shows me two things. It either shows me, wow, there was lots of variation because the large standard deviation that went into this, or it was only based on a few individuals, so it's also not very accurate. So there's kind of like lots of room for error in what I'm saying. Now in sample 2, we could either have a small standard deviation, which would give us smaller standard error bars, or we could have a very large sample size. And with that large sample size, that would also make us more accurate. Like if I'm looking at two samples, a sample of like people sitting in a field having a picnic versus like a sample of like a college lecture hall, and I want to know like the average height of a college student. In sample 2, it's based on way more individuals. I can say with more confidence that the average height of a college student falls within this range of sample 2. Versus sample 1, I only have, 6 people to base my conclusions on. I'm less confident. There's like, I have larger standard error bars because I have less samples to draw my conclusions from. Okay, so let's kind of see what this means as far as how it applies to like our class. So when we look at standard error bars, basically, like I just got this one from Google Images, But if we pretend that each of these 3 bar graphs are representing fertilizer added to plants, 3 separate fertilizers, and I'm curious if they support plant growth. So in population 1, I see that the standard error bars between the before and after treatment, they overlap. So like right here, there's an overlap between these bars. So what this means statistically is that there was no significant difference on plant growth with fertilizer number 1. And that variation I'm seeing, because if I look at the average after treatment right here, I might be like, oh yeah, the average was higher. But if I include my standard error bars, that average being higher could have just been due to chance. What if the plants just had more sunlight or something? And so, or genetically grew bigger, who knows? But it wasn't due to the fertilizer. So when there's overlap, statistically speaking, there is no difference. And that variation, that difference is just random and due to chance. Now, if I look at population or treatment number 3, I look at the before and the after, you can see how there's no overlap in these standard error bars. And what that means is that there was a significant difference. Fertilizer option 3 really statistically significantly affected plant growth. And those plants did grow taller with that fertilizer, all because the standard error bars do not overlap. So when I look at how this may be asked on a test or something, so this is an FRQ from 2016, and it wanted to know, oh, you can't see part B down there because I don't have it recorded, but basically they wanted to know if the amount of prolactin in the blood was affected by exercise. Or like, so they did at 0 hours, like took the measured prolactin beforehand, and then they exercised, and then an hour later measured prolactin. And what you can see here is even though after exercise, the mean or the average is higher, the standard error bars overlap. So you would actually say that there is no significant difference in prolactin release with exercise. Now also, there's also no significant difference in prolactin release without exercise because these error bars overlap. Now, when we look at how you would draw these on your own though, so here's another FRQ from 2014 or 15 where they are asking students to create a labeled graph to show the 3 populations sample means within 95% confidence. 95% confidence means plus or minus 2 standard errors of the mean. So here they've given us a standard error of the mean as 1. Now sometimes standard error of the mean might be 0.5, might be 3, it could be different numbers. Here they were trying to make it easy for students. So when you look at how you would graph this, here our population 1, our mean was 9. So when we graph it, here's 9. Now it says plus or minus 2 standard errors of the mean. So if my standard error of the mean is 1, that means I'm going to start at 9 and I'm going to go up from 9 to 10 to 11. You have to add 2 of them. Then I would go minus 2. So I would go start at 9 and go from 8 to 7. So on this, I have plus 2, minus 2 standard errors of the mean, and that shows with 95% confidence that the actual population trichome density falls within that range. Now, if I look at sample 3, same thing, the average or the mean was 11. So plus or minus 1, 2, that brings me up to 13, minus from 11, 10, 9. So here is my 95% confidence that the real population falls within that range. So now, when we look, and et cetera, same thing with the last one. But here it says, identify the 2 populations that are most likely to have a statistically significant difference in the mean. That's going to be population 1 and population 3 because their standard error bars do not overlap. Therefore, there is a significant difference between those 2 populations. Whereas population 1 and 2, no significant difference. Between 2 and 3, no significant difference. Okay, I hope this was helpful.